Thank you very much. Hi, and also just want to start by saying thank you for the invitation, Taufik, and to the organizers for the work to organize this meeting. Your introduction yesterday really resonated with me about the way I felt when I came to my first meeting on human neuron recording. It was also in 2014. It was the other meeting for the human single neuron meeting in Johns Hopkins, and I distinctly remember one of the first people I met at my first meeting where I was presenting was you, and you were so warm and welcoming and encouraging to us, even though we hadn't done anything yet. It always sticks with me when I see you. I think of that fondly. And also, it caused me to look back at the presentation that we gave. So part of the goal that I have for my talk today is to do a little bit of reflection, retrospective, and how we're doing, and remarking on the incredible progress that everybody here is making in the techniques and the approaches and the findings. It's really, to me, exciting to see how far we've come, but also to be a little forward-looking about some ideas, maybe a little bit out-of-the-box ideas of where we could go. And so that's the goal for today, stimulate some discussion. I know we have a panel discussion later, so hopefully this will feed nicely into that. And even that long ago, there was this book from the organizers of this other meeting where in 2014 they were predicting that brain tissue from surgeries would be, it wasn't used heavily at this time, but it will be much more heavily used, and people will be doing these patch clamp recordings and making incredible progress over the next decade. And then I believe I gave a joint talk with Ed at this meeting. It's probably your first meeting for human neuron recording, and found this slide, and I was like, oh wow, this looks really great. I don't think we actually knew that we were going to be recording this sort of triple modality data from single human neurons. At the time, patch-seq hadn't even been invented yet. It was a year later. The first mouse cortical cell type taxonomy hadn't been published yet. That was a year later. But kind of the inception of the ideas sort of was there, that we need to get these multimodal features to really make sense out of this sort of data. Fast forward to where we are now, fortunately for us, it worked out pretty well, and just from an Allen Institute-centric standpoint, now this has just become routine, where we're doing patch-seq recordings. We're trying to get single-cell morphology, physiology, transcriptomes with mapping to a taxonomy of brain cell types in not only the human, but the mouse, the macaque, the human, as you heard yesterday from my colleagues, and integrating viral targeting as well. So I think we took it even a little further to make them look really good about their prediction. And also, one of the most exciting things in those early days for me was that, and as many of you have observed too, is that it's just kind of crazy how long this human surgical tissue can last sitting out on a counter. Looking back, I was probably thinking, like, when do I get to go home? Like, this tissue just will never sputter out. And we did a study where we showed even 1 day, 2 day, 3 days sitting out on the counter, if you just constrain bacterial growth, that you can get recordings that look very reasonable, very similar, and we quantified some electrophysiological properties. And we showed that that was sort of the predecessor to knowing that this is going to work when we move these into cultures. And it was very clear that the goal was move this into longer-term cultures so that we could get access to the molecular genetic strategies that have really advanced our understanding for rodent cellular neuroscience. And simple demonstrations like that you can virally express within a couple of days for channel rhodopsin or GCAMP in human neurons. Our team, but also other teams and pioneers here were showing that at the time. And even looking back as far as 2014, I forgot that we had done this at that time, but we were even exploring adeno-associated virus, trying to figure out how to get it to express faster. As you probably know, in a mouse experiment, you inject in vivo. It may take 2 weeks, 3 weeks to get peak expression. But we were trying to figure out how to do this in just 2 or 3 days. Even we didn't have many cell type specific enhancers or promoters at the time that we could use. In this example, I'm not even sure if those are really neurons or not neurons, but I think that's sort of the point, is that in that early stage, looking back, it seems like improbable that we were going to get to triple modality patch-seq experiments. Sounded science fiction, and seemed improbable we were going to be able to do cell type specific targeting and genetic labeling in human surgical tissue. But now we've had some pretty nice successes, and my colleagues Brian Lee and Brian Kalmbach introduced some of these studies. So, since some of them are published, I don't want to go into great detail, but just to say the thing that really hit home for me, that this is now a firmly established paradigm for rapid viral genetic labeling, patch-seq recording in the human brain slice cultures, was the study where we did targeting of GABAergic neurons. Something so simple just as cell class like GABAergic neurons got us sampling of virtually all of the transcriptomically defined GABAergic types over many years of data aggregation. And this was a study with over 1,000 recordings. With quality control, that number went down to 778 neurons. But in doing so, we could see two really important things. One was for a patch clamper, we can see this labeling within less than 2 days with this exemplary unicorn virus that we call DLX 2.0. And it's 99% GABAergic specific, and start to see sampling of specialized cell types in the primate brain, even also rare cell types, although double bouquet cells shown here are not as rare, but you certainly can't study them in a mouse brain. So lots of other interesting nuggets in that study, but the main point is just thinking about where we are now and what is the next thing that's sort of in its infancy and that we need to kind of invest to move the field forward. My take is that in the last 10 years or so, a lot of the teams here have been investing in building the platforms and the capabilities, which is our foundation, but not as many people have advanced to sort of implementation of the platform for real, rigorous biological discovery, although we're hearing it even in this meeting, like talks that are really spectacular, that it's definitely happening. For example, instead of showing that it's possible to do this as a one-off methods paper, now they're turning into high-profile studies. The work that's going on in Berlin, the team, it's really spectacular. So looking across the field too, I'm very excited. Just two examples from the recent literature. One is the AAV cell reader vectors from Josh Huang and Derek Southwell. They're getting pretty close to an orthogonal strategy that can pair with cell type enhancer viruses. It's a different approach to cell type labeling where they can drive opsins and do, in this example, they're using a cell reader for CalB2 neurons, which is a surrogate for VIP subclass in these human slice cultures, and that they're showing they can do optogenetic connectivity mapping and dynamics. It's not to the level of the group in Berlin, but I think in a few years of further development, this is coming along nicely, and it could potentially reach that level. Similarly, I was thinking about why there aren't more people using this platform for gene function analysis, where you can do direct perturbation and see what's the effect instead of these sort of correlations between genes and properties that we sometimes get stuck with patch-seq data. We should be able to apply these methods for either AAV CRISPR in a cell type specific manner, or in this case, shRNA in an AAV vector. This seems to work really well for robust gene knockdown, not quite maybe to the level of the robust effects that we want, but on that trajectory that you can see that that's coming. But for today, the main thing I wanted to present about is definitely down the rabbit hole and talking about patch clamp recordings from neurons in postmortem brain slices. And it's something that our team has attempted in spurts and given up and been very discouraged. It's been very hard, and just keep coming back to it and can't kind of get away from it. And I blame Hyde, because you're the first one that showed me that this was possible in human slices. But even going back to 1998, there was a paper, and I bring this up a lot. I think people forget about this paper, but a group had showed that you can take a young rat brain and just decapitate the rat and leave the head on the counter to passively cool for up to 5, 6, 7 hours post-mortem delay, then go and make brain slices and do patch clamp recording. That's not how I was taught that you do patch clamp recording, right? It goes against every bone in your body of how you're supposed to treat that tissue. But they showed, beautiful examples, and I think maybe some people just ruled that out as couldn't be true. And later, you and your colleague published this figure in this book chapter, and definitely a bigger challenge to work with these rapid autopsy, short postmortem interval human tissues from diseased brain donors. In the normal sort of acute brain slice, you can get the normal beautiful recordings, and I think in your case up to 3 hours there was some signs of repetitive spiking, and definitely they're alive and synaptic events, but longer than that was really not so good. So I don't want to bore you with lots of bad looking recordings, but just to say that we see over the years when we tried this off and on, we've seen just this over and over of tissue that had even 2-hour postmortem interval, sometimes as long as 6 or 8 hours, but every neuron shows signs of like anoxia, ischemia, like you get a single spike, you don't really get repetitive firing, doesn't look so hot. So that's really bothered me. I feel like that it could have to do with just the types of tissue that you're working with, and if you had the right kind of brain to work with a low postmortem interval, that it should be feasible. So we've been sitting on this unpublished data, but I think about this often and have picked this back up again. We'll see if it's worth it this time. But I was doing experiments to try to test this idea. If you did this under more controlled conditions, how well could this work? So this is adult 6-month-old mouse brain, where we did the same thing, anesthetize with isoflurane and decapitate, leave the head on the counter. Don't want to put gory details, but that's what we're doing, and just letting it passively cool. Then take the brain out after either 1, 3, or 5 hours, again, like the original study. And yeah, I mean, not only can we replicate the beautiful results, but here the point was to show that cell type specific electrophysiological features are preserved. These are blue layer 5 ET neurons in the cortex, and red are the fast spiking interneurons. And of course, as we heard yesterday, like you would expect, these fast spiking interneurons that have high metabolic demand would be the first thing to go. So I was really shocked and impressed when I saw that. These are just an example. We can record these. It's not like a fast recording and it died. You can record them for long times and run your whole battery of protocols, and they look really beautiful. The cell type specific signatures are clearly there. So this time, I thought, that's where we got stuck for a long time. I thought, why don't we try a different strategy of trying to advance this stepwise, instead of just using our precious human surgical tissue and getting it from 98-year-old dementia donors, which is all we had access to, is to move through first macaque, where we have whole brain from our tissue sharing program, well-controlled conditions, and see if that works under similar simulated conditions of postmortem, just like a human donor would be. And then try human neurosurgical tissue resections right before you go to postmortem diseased brain donors. So the methodology is similar here. Just want to emphasize that brain tissue collection, 2 hours is the time point that I focused on, because I think that's become really feasible for the human brain donation. And passive cooling at room temperature, there's no carbonation, and then at that point take out a block that's about a cubic centimeter like we normally do, and make brain slices, culture them, virus label them with our AAV cell type vectors, and image or patch clamp recording. So just an example of an unperfused, we've done it with perfused or unperfused macaque whole brain or hemispheres. This is the region we focused on to start, the superior temporal gyrus, and like an example of a human neurosurgical tissue block as well, and the slices and the viral application. To make it visually appealing and simple, we tried to apply what we thought was a hard test case of brainbow or AAV TETBO labeling to really demonstrate the extent of labeling that you can get. This is not like something where you see one healthy neuron in a sea of dead neurons, or at least I need to convince you of that. So this is our first set of results for 2-hour postmortem simulation in macaque with the whole hemisphere or whole brain, and the brainbow virus combination just pipetted on top. We can see hundreds or thousands of neurons labeled. This is as good as our normal regular brain slice culture, right? I don't understand it. We replicated this in 4 separate macaque cases. We replicated it for multiple regions of the cortex. We replicated it for subcortical regions, and it worked well every time. There was no failure. So this is a robust result, and reproducible. And then finally, to make it relevant to this meeting, really trying to hustle in the last few months to get to convince my colleagues to let me mistreat this human precious surgical tissue. And so a few cases where we had very small, like trimmings almost of what you would normally get, could go forward for this sort of pilot work where we leave that tissue for 2 hours and then do the same thing. And here's the result for the first and second human cases. We can also replicate that exactly. We can also apply other vectors for pan-neuronal labeling or for GABAergic specific labeling. We're not losing our signatures of cell types or our ability to mark them. And finally, that recently trying to get some recordings to really show that you can take it all the way. This is from the pan-neuronal labeling in the human culture. So 2-hour postmortem, make the brain slices, virus label, and now 7 days in culture, 7 days after the virus infection with the AAV vector. We can record a variety of different types of neurons with different intrinsic firing properties that resemble similar kind of diversity. We don't have patch-seq in this case for this pilot, and I'm still in the process of sort of trying to do more rigorous feature analysis, but that would be the next step. Surprisingly, you can fill these neurons and still see beautiful dendritic spines. You can run the normal protocols. You can see lots and lots of spontaneous synaptic activity. So that's for the glutamatergic neurons. Same thing was done for the GABAergic neuron targeting. Because it's a sort of pan-GABAergic virus, we expect a lot more diversity of firing patterns, but certainly we can pick out, like, yeah, this is the most prototypical, like, fast-spiking interneuron, like a parvalbumin subclass, and some others that may be more consistent with, I think this might be a VIP pattern, or this one has a signature that we see of the ramping of the memory potential, like a chandelier cell. That happened to be an unlabeled cell that we filled with Alexa. So, you know, still, the case is not that nothing is changing, right? But it's just saying you can still recognize these canonical cell types by their normal intrinsic electrophysiological signatures. And yeah, the next step would be to characterize to what extent those are changing. And to take that example of the fast spiking interneuron that fires like crazy, right? We do some, I can't explain this, like how is this possible? You give a 10-second step depolarization, this cell is continuously firing for 10 seconds at 130 hertz without exhaustion after this sort of abuse. And you can see the crazy amount of spontaneous mini excitatory synaptic events over a long 30-second sweep. And then, you know, just starting to get into, can you do functional perturbation? We put in synapsin channel rhodopsin, like the most basic thing you can do. We can see beautiful labeling. We can find virus-labeled cells. We can evoke nanoamp-sized photocurrents that would be consistent with ability to use that for connectivity mapping. And then finally, very recently, maybe many of you saw this study that was published about vitrification of rodent brain slices, which is a process of taking the tissue through cryoprotection and loading of a special formula of cryoprotectant, dropping the tissue temperature down to like liquid nitrogen storage, and then bringing it back and unloading the cryoprotectant. And this group, German et al., showed that you can get recovered post-vitrification slices that have relatively impressive looking morphological preservation. So this is the CA1 region of fluorescently labeled cells before and after vitrification. And recordings from those CA1 pyramidal cells, you can see that they're recovering clear functional repetitive spiking. Again, you know, characterizing the extent to which it's changed. There are some subtle changes there reported, but it's pretty strong. It was really impressive to us. Synaptic events, even LTP after vitrification. And we started working on this with collaborators to try to replicate that and extend this, and using even our own special formulation as a collaboration with a cryobiology expert, Matt Powell-Palm. I'm only showing you just our exciting very first recording that we did. This is what the brain slice looked like from a mouse after the vitrification. So this slice was taken down to minus 110 C, immediately taken back out and put on the patch clamp rig. And I don't think that our team, if they didn't know, would know that this was just not a normal slice from our normal mouse pipeline. There's the cell that was recorded and really normal looking pyramidal cell features. So of course, you know, we've done maybe a dozen recordings in this one day of first experiment. We're very excited to push on this, to apply this for human surgical tissue. or even macaque, and see, I mean, I expect this to be species agnostic. And so if you put those pieces together, I don't know how much of a stretch this is for all of you, but for me, just having this epiphany of these types of things that we're seeing now are possible. If you put those pieces together, this potential ability to get really low post-mortem interval human disease donor brains through like the medical aid in dying type scenario, and ability to make brain slices that have good functional properties immediately, but then also to be able to do things like vitrification and make like a little mini brain slice bank. And you could be pulling those slices out the next week or maybe 10 years from now. It should be indefinite storage, and so that will be very exciting to see where that goes. This leads me to this sort of what I think is a roadmap of the tools and taxonomies, spatial atlases, the viral tools that we've made, mapping tools, as Ed introduced in his first talk yesterday, the types of things that we can do with prospective marking of rare cell types for targeted recording, or channel rhodopsin-assisted circuit mapping, or gene function perturbation studies, and specifically now maybe a way to not do disease modeling, but to actually study the human disease brain directly and to use functional type analyses like patch clamp recording. Maybe this will save the jobs for patch clampers forever, right? We'll see. That's my hope, that we're not kicked out by AI. So I'll bring this slide back for the questions, but instead of going through it, but just want to end by thanking the people that helped me, especially Brian Lee and Brian Kalmbach, and some of their team members, Scott and Chameleon, for believing that this is worthwhile to try to push this avenue on the postmortem simulation. And thank my team members and our collaborators, Matt Pompowell and Tony Consiglio, and our surgeon, Charles Cobbs, who provided those specimens for these pilot studies. Thank you very much.
Thank you, Jonathan. It also opens the perspective of going beyond cortex and in post-mortem tissue, so that would also be very exciting. Yeah, great stuff. Questions? There's a question over here.
Thank you so much, Jonathan, for this talk. So I wanted to ask you, we see differences between different brain areas in primates, and we haven't tried to look at whether the time, post-mortem time, affects the times that the slices are, that we can record from the slices. But your protocol seems to fix these 2 hours or going into, is there any difference, or is there anything that you can think about that if you're doing V1 slices or hippocampus or prefrontal or temporal lobe, if that is going to be different from these different areas?
I think that's a great next step. I mean, one of the things that we would really like to do is to extend this sort of logic of, maybe you heard from Ed, the theme of trying to study selective vulnerability of cell types in disease. In this paradigm, I'm interested in understanding what's the selective vulnerability and resilience to the postmortem interval for different cell types. And I think that would be a logical next step. You know, you might predict CA1 pyramidal neurons might be much more vulnerable because of high NMDA receptor, high reactive oxygen species generation, that kind of a thing. I mean, I've done lots of recordings where there was like anoxic depolarization and irreversible loss of the response when you overactivate NMDA receptors. So obviously, it needs much more work. It's really sort of very small pilot at this stage, but yeah, I'd be very excited to chat more about ideas of how to advance that.
Thank you.
I mean, I remember we had lots of conversations about, you need to do this like really carefully just for the normal slice prep, right? Like, I think that's a common experience people have getting into their first human specimens is, it's got to be 5 minutes from the OR or 10 minutes, right? So we feel a little bit more relaxed, but you know, this is a very specific niche use case.
Next question. You had a question, right?
Body for like 2-3 hours. Are there other processes like on a systemic level, pH or blood flow, rigor mortis, that affects the tissue in a different way compared to if you just cut it out and leave it there?
It certainly could be different. I feel like that was the goal of using like whole macaque brain, was to get closer to that scenario where you at least have a whole intact brain. but it's still, you know, a little bit different than the actual context. I think we'll have to try it. But the main thing is that this is without doing anything special. Like, there are still possibilities to apply other special solutions, rapid cooling, other things that could make it even better than what I was doing, which was very crude.
And I had a question. Similar? Okay, then the last question is for Frans.
Thank you for the great talk. This is really interesting stuff. So my question is regarding the temperature. So for this pilot in human right now, you're leaving the post-mortem or the resected tissue at room temperature. Is that correct?
Yes.
For the interval.
Yes.
And then the follow-up question would be, what is your intuition about temperature? Because as you know, we do this transport business where we transport tissue from remote sites, sometimes for 6 hours, and we have a similar experience that we can do perfect recordings from that tissue, but we found that temperature is really key. So if you transport it as a block, we find that we should cool it to something like 6 degrees Celsius, whereas if we transport them as slices, we have to keep them at room temperature, because if we cool it too far, then the cells won't recover after that. So that would be my question, what your intuition is there, how important the cooling aspect would be, and whether that would be something that's applicable to patients that have just died. Could you maybe cool them?
Yeah,
like,
that's a
aggressively?
great idea. I think that's sort of the way to go. I don't have the answer to the question, but I agree with you. That's going to be very important to do any sort of serious cell and mechanistic studies in this context from human disease brain. I'll just end on a funny note to say that our experience is that other people feel this is very important how you treat that tissue in that period. And we have colleagues like walking by the hood and seeing just a block of, or a whole macaque brain or a block of human brain, and like, just they really want to intervene and do something. Like, it's like, no, don't touch that tissue. Like, leave it alone.
With that, thank you very much, Jonathan, for a great talk.